Good morning, good day, good evening, wherever you are. This is the fourth session of the grand jury investigation with a real judge, real lawyers, real expert witnesses, and real witnesses outside of the system.
So my name is Deanna McLeod, and I am the principal and founder of a medical research firm called Kaleidoscope Strategic. I have a background in immunology and psychology from McMaster University, which is the home of evidence-based medicine here in Canada. My particular perspective is unique in that I worked in industry for 10 years in many roles in medical marketing and sales. And I became concerned at the end of about 1999 by a trend that I saw in pharmaceuticals where benefits were emphasized and risk minimized, both in how they conducted their trials, both in how they conducted their marketing, and also their business practices. So in 2000, I launched and founded an independent medical research firm that was designed to help clinicians prepare objective clinical guidelines. So we provide research, medical research, writing, administrative support to help that. So what we're doing is we've worked with hundreds of doctors in Canada to prepare guidelines in oncology. And we spend a lot of time looking at clinical trials and clinical trial design. And one of the unique perspectives that we have is we've acquired over the years an ability to see how pharmaceutical companies manipulate data. And so when I'm presenting, what I'm doing is I'm bringing our firm's many years of experience in preparing these guidelines to look at specifically the Pfizer phase 3 trial, and particularly the 6-month publication or the 6-month follow-up data for that particular trial. And I'd like to highlight a number of things that would make me question whether the reported benefits and risks are actually accurate. So moving on to that, one of the reasons why I'm choosing to look at the phase 3 clinical trial for the Pfizer data is that here in Canada, Pfizer is the backbone of all mRNA vaccines. And although there is also use for Moderna, it's much more limited. And Pfizer is the vaccine that's being promoted for children. And what I wanted to do, because this particular trial is the backbone trial, meaning that all the other trials are built on this trial, I wanted to make sure that in scrutinizing this data, that it was that the benefits and risks were actually fairly reported. And so that's what we're going to get into. And in Canada here, one of the other things too is that there's a lot of discussion, after this phase 3 trial was completed at 2 months with 2 months follow-up, and they began the mass vaccination rollout. They then shifted to observational studies. And observational studies being real world analysis of data that basically said, this vaccination rate is this high and our numbers were this, and we looked at COVID-19 cases, et cetera. But that type of data is actually fraught with bias. It is very difficult to interpret correctly. And so our firm basically sought to look at exactly the level 1 data, because that is the only way that you can actually create a causal link. So if you have an observational trial, you can say it's associated and there may be a benefit. But the only way to actually prove something in clinical trials is in the context of a phase 3 trial. And so proving efficacy or proving safety. And that's what we're going to look at today. So I know that my colleague Vanessa mentioned some of the strengths and limitations of this particular trial. But one of the things that I want to highlight is the fact that it was conducted in healthy individuals, which is not uncommon whenever you're thinking about a vaccine trial, because what you want to do is you want to treat the healthy in order to minimize transmission to the more vulnerable people in the society who might not be able to mount an appropriate immune response. However, if the vaccine or the purported vaccine, I'll call it an inoculation, is unable to stop transmission, then it actually isn't a vaccine and should be studied in actual population that it is meant to benefit. And in this case, the benefit of the population at risk was specifically elderly people in Canada in long-term care facilities, specifically with multiple comorbidities. And in addition, one of the things that we found when we did a Royal Commission report in Canada was that these patients were also had the majority of them had documentation that said that they would have no medical intervention. So it's a wildfire where you have a disease that transmits through community spread. You have elderly patients who have no strong immune systems. All that we have under-resourced facilities, and we have a virus, and then we have people who basically have mandates that say no medical intervention. So you can imagine that in Canada, the death rates were very, very high. In fact, 81% of the death rates were in people who were in long-term care facilities. So studying treatment, which we'll call an inoculation because I don't really think it qualifies as a vaccine, in a population of healthy people does not help us with the problem that is specific to long-term care facility transmission in elderly people who are immunocompromised. And again, I believe my other colleagues mentioned this, but the vaccine, the inoculation should have been compared to the standard of care if we wanted to prove something. And the standard of care was natural immunity and treatment. It was not placebo in the sense of non-treat or somebody without any natural immunity. So we should have basically compared those two things rather than comparing it to a placebo. And based on the trial design, we cannot say anything about whether the inoculation is better or worse than natural immunity because they weren't compared. In terms of the testing, it's also been mentioned previously that it was selective testing. They did not systematically test they did not do systematic testing in the sense that they waited for somebody to be symptomatic, and then they left it at the discretion of the investigator to actually test or not to test. And what that actually did was it created an investigator bias. And I think my colleague also mentioned the fact that they were taking treatments to lower temperatures that might have minimized symptoms. There might have been infection without symptom, which might have been compromised the efficacy endpoint in that regard. But they also weren't able to detect asymptomatic infections. And also, they didn't use the standard virological test. So, for instance, when we're looking at clinical trials, we want to make sure that whatever test they're using to determine the efficacy endpoint is validated. And the standard for testing viruses is a virological assay, and that was not used. So when we look at a trial like this, we immediately begin to say, is this a manipulated endpoint? And that's what we begin to question. Further to that, let me just keep going here. The endpoints for the trial, and again, I'm going to focus in on the primary endpoints because the primary endpoints are such that the study is actually designed to detect statistical significance in those particular endpoints. One of the endpoints was, of course, COVID-19 symptoms plus a positive PCR test 7 days after the second dose. And the second one was safety data. So the safety data, there were different forms of safety data collected. One was solicited, and that was the reactogenicity data. And they only looked at a subset of patients. They only tested that the reactogenicity in a subgroup of patients, not the whole trial. And they only did it for 7 days. And then they had unsolicited safety. So that means that a patient could say, I'm not feeling well, and they would report it in an open diary, and then they would mention it. And if it was a severe any type of adverse event, unsolicited adverse event, that it would basically be reported for 1 month only. And if it was a severe or serious outcome, it would be reported for 6 months. So you can imagine that if you're reporting the endpoint, continuously monitoring the endpoint, but only measuring or monitoring the safety data for 7 days or 1 to 6 months, then what you'll miss is a lot of the safety data. And so there was inappropriate safety monitoring for this particular trial, especially when you're considering that you're using a genetic therapy in a population of healthy people. They also focused on clinical rather than subclinical endpoints. And when I'm talking about subclinical endpoints, that means that they didn't look at biomarkers and different factors. So they could have been looking at D-dimer levels, for instance, if they were suspicious that thrombosis might be an issue. And the other thing, too, is that the secondary endpoint was severe COVID-19 symptoms. So although we're claiming that it reduces both COVID-19 cases and severe COVID-19 cases, the other one was a secondary endpoint, and the numbers of events were insufficient to actually establish causality. Finally, I'd like to talk about the fact that really what we want to be looking at is all-cause morbidity and all-cause mortality. All-cause morbidity means looking at the sickness from the disease, COVID-19, as well as the sickness from the actual vaccine, both looking at the symptoms and the symptomatic burden for that, and also for the death related to that. And of course, the particular the trial reported de-emphasize that and emphasize something that really isn't clinically significant, which is symptomatic positive PCR tests. And the reason for that is that the majority of those symptomatic cases were mild and really not of a concern. What we really needed to know was whether it was going to stop COVID-19 hospitalizations and deaths in elderly people, and whether the all-cause morbidity and mortality was higher in the inoculation group versus the placebo group. So, and I guess the other final thing is, and we mentioned this earlier, is if it's going to be presented as a vaccine, then we would have to be able to prove that it stops transmission. And this was never part of the clinical trial design. And so therefore, anybody who's claiming that it stops transmission would be making false claims. Anybody that claims that it reduces severe COVID-19 disease based on this trial would be, again, making false claims because the number of events weren't sufficiently high. And anybody who's making claims regarding lowering deaths, again, would not be supported by this particular trial. So I did want to emphasize the fact that after 2 months, we had a crossover. So there was an unblinding of the two groups. And when they were unblinded, people in the placebo group were offered the opportunity to cross over into the inoculation group. And about more than 80% of the people, I believe it was close to 89% of the people, actually crossed over. And what that means is that as of 2 months, we no longer have efficacy and safety data from a controlled trial. So all the safety data that we're going to be looking at, and that's the angle that I want to be looking at specifically, is really obfuscated by the fact that now most of the placebo group have actually been inoculated. And so when we present the data, we'll be looking at the unblinded phase, which was really only 2 months of the trial. And we don't actually get the full benefit of a randomized clinical trial to the point of 6 months, which is this data, because of this crossover. And I don't want to ascribe intent. However, there is a great benefit if you're interested in minimizing safety to have your placebo group cross over at a very early stage. Therefore, you will never be able to have a causal link to long-term safety issues. When it comes to efficacy, one of the things that we noticed in this particular trial report is something called combined efficacy endpoints. So one of the things in this particular trial, as they noted in the discussion, was that the immunity was waning at about 4 to 6 months in the adult population. And this is a publication that presents 6-month follow-up data. And in this particular publication, what they did, instead of reporting adult outcomes as they should have as a follow-up to the original 2-month report, what they did is they combined the reporting of the efficacy outcomes of adults and adolescents. And the adult outcomes, of course, had 6 months of follow-up, but the adolescents had 2 months of follow-up. And by combining these two together, what they did was they boosted the numbers overall so that they could continue to report a 90%-ish efficacy rate. And that was likely due to the boosting of the younger adolescent group, which was basically only followed up for 2 months. So we actually, at this point, do not know what the efficacy at 6 months was for the inoculation in adults. That data was not provided in the particular report. So one of the other things that I'm going to do here is I want to compare. What we did was we pulled the data from the supplements and we compared it to how they were presenting the data for efficacy. And we used the same treatment using relative risk reductions and absolute risk reductions, as Vanessa had noted previously. And we put them in one table so that you can compare the efficacy versus the risks of this particular vaccine by looking at it in one particular table. So here you'll see for symptomatic cases, which is again our primary endpoint, there was a net reduction in the number of symptomatic cases from the inoculation group to the placebo group. And there's our beautiful 91% efficacy. Remember that this is both adults and adolescents combined. It's not adults alone. And what that does is it provides about a 4% absolute risk change, which is over here, minus 4%. So there is a purported benefit, but if you're inoculating 20,000 people, a 4% benefit is not that great. It's not dramatic. Here, it looks much better whenever you're looking at relative risk change. And that's why they tend to emphasize that, because that looks much more impressive than the 4% benefit that you're getting when you're looking at an absolute risk change, which is the number of people who are actually benefiting from the inoculation. And what I'm going to do is I'm going to just jump right here to the other primary endpoint. And these are all primary endpoints. So treatment-related adverse events, severe adverse effects, and serious adverse effects are all primary endpoints of this trial, which have not been mentioned to the same degree as the efficacy. And one of the things you'll note here is that in the inoculation group, the investigators ascribed adverse effects, which when you looked at the reactogenicity data are actually COVID-like symptoms, to 5,241 people who participated, and in the placebo arm, only 1,311 for a 300% increase. And if you consider that the absolute risk increase was minus 4%, that's the benefit there, the risk is plus 18% here. So there's more people at risk with this particular inoculation than you have people benefiting from the inoculation overall. When we look at severe cases here, you can see that there's a net difference of 22. This number of events is clearly not clinically meaningful. 22 difference is not, but although if you put it in a relative risk change setting, then it looks like 96%. However, there's only a 0.1% benefit overall to reducing severe symptomatic COVID cases. So again, an overemphasis by looking at the relative risk reduction and a very modest or minor benefit overall. However, when we look at severe adverse effects associated with the vaccine, you see that in this group, you have 262 versus 150. So that's a difference of 100 cases, severe adverse effect cases here, and an increase in 75%, and it's an absolute risk increase of 0.5%. So again, what we're seeing here, again, when it comes to severe cases and severe adverse effects, that you have more risk than you do benefit for this particular inoculation. When we look at serious adverse events, which are very concerning to me because we're treating very healthy people, and a serious adverse event as defined in this particular study is an event that requires inpatient hospitalization, is life-threatening, results in death or persistent disability. And you can see once again that you have more severe adverse events, 127 versus 116, in the inoculation group, which is an increase of 10%. And the incidence of these is about the same as the risk of many people in getting COVID-19 overall. I mean, it's small, but significant when you're considering how severe those adverse events are. If we look at, so that is as close as we got to looking at all-cause morbidity versus benefit. And here, when we're looking at deaths, again, we went to the supplements table and we pulled the deaths, and these deaths are basically from the unblinded phase only, which means it's the first 2 months of the actual trial, or the first 2 months of follow-up from that actual trial. Because they crossed over, then it's more difficult to find out. But in this unblinded phase, the deaths were comparable, 15 to 14, inoculation to placebo. However, we have to remember that this is a very healthy population with their diseases that were controlled, with controlled disease. So to see this number of deaths in the placebo arm where they actually caught COVID-19 might be reasonable within a 2-month time frame in 40,000 people. But to see this many deaths when people are purportedly not getting COVID-19 but are getting inoculated is concerning to us. When we looked at the deaths after unblinding, you can see here that there were 5 deaths in the unblinded phase. Oh, sorry, excuse me. I'm missing a number there. There were 5 deaths in this case and 0 deaths in the placebo arm, for a total of 20 in the inoculation arm and 14 in the placebo arm. So again, just to be clear, after the patients who were in the placebo arm crossed over and received their inoculation, there were an additional 5 deaths. For a total of 20 deaths in people who were inoculated in this study and 14 in the placebo. Again, we have to consider that at least in this particular case for the second half, there were more patients who had gotten inoculated than the placebo group, but still this is concerning. This is not the trend that we would like to be seeing. And finally, if we actually look at COVID-19 related deaths, there was really only a difference of one death between the inoculation arm and the placebo arm. However, when we looked at cardiovascular deaths, we saw that there were 9 cardiovascular deaths in the inoculation arm and 5 in the placebo arm. So again, what we're seeing is if our desire was to see morbidity and mortality decrease in the target population, which was elderly people, what we're seeing in this particular study is an increase in morbidity in the sense of risk and mortality in a healthy population. And so we are very much familiar with cancer research. And if we ever saw something like this in cancer research, we would not even proceed with administering this to people who were in the end of their life. So these results are extremely concerning. And I'm just going to touch base very briefly on the companion trials that were conducted for children. And so one of the things that they do is they create a phase 3 trial, and then they basically do these companion trials where they, in this particular case, they're called immunobridging trials. And these immunobridging trials, basically, we're just focusing at antibody, neutralizing antibody titer production. And what they did, rather than doing a clinical trial, if we were to be thinking about the people who are at greatest risk, it would be children. They're at no risk of severe disease. Therefore, what we would have liked to see is the safety being much higher, the much more rigorous, larger numbers of people enrolled, very strong endpoints like clinical, subclinical safety, clinical safety, long-term safety, morbidity, mortality. We would have liked to see all of that. However, the trial that was designed, or the endpoint that was used to approve these vaccines or these inoculations, was non-inferiority of the neutralizing antibody titers, which what that means is that they compared antibody titers in people who were 12 to 15 years, and they compared them to antibody titers in 16 to 25-year-olds. So this study wasn't even designed to make any claims regarding clinical efficacy or safety. And anybody who makes clinical efficacy or safety claims based on this study is basically misreporting or making false claims. Again, they did the same thing with 5 to 11-year-olds, again, a slightly smaller dose, and they compared them to the neutralizing antibodies of 16 to 25-year-olds. And if you could note here that the number of patients or people enrolled in each of those 2 cohorts is very small. And so this is the basis by which we're moving forward with the vaccination of children. Now, they did, enroll 1,000 people in the clinical side of things, and in the placebo side of things, about 1,000 in each arm. And these particular endpoints, unlike the main trial, are descriptive endpoints. So they're not meant to actually claim anything, but they do give us some sort of a window into, the benefits and risks of the inoculation in this particular group. And I'm just going to very quickly move to this side, because this is the absolute risk change. And this is the one that we really need to keep an eye on when we're looking at absolute clinical or clinically meaningful benefit. For symptomatic cases, it was minus 2%. So there was a reduction based on this. And if you can note here that it's really only a difference of 15 cases. And we also have to note that symptomatic cases in children are really not clinically concerning. So I would probably move to say that this is a clinically meaningless endpoint. Again, they're at no risk of severe disease. And of course, there was no differences in severe disease. So that's not a benefit for them. And however, if we look at treatment-related adverse effects, what we see is this concerning trend again, where we have more risks or more adverse events in the inoculation arm than the placebo arm. And I'm just going to emphasize this very carefully. The types of adverse events that were reported in the placebo arm and the inoculation arm are very similar, and they're clinical symptoms. They're COVID-like symptoms for the most part, when they were reported, at least based on the reactogenicity data. To see more COVID-like symptoms in people being inoculated versus the people who are actually getting COVID-19, according to this particular data, is very curious. And again, that's a 1% increase in adverse events here. But when we look at severe adverse events, remember, there's no benefit here. And suddenly there's an increased number, although small, of serious adverse events in the inoculation arm for a 0.4% increase. And again, when we look at the serious adverse events, and remember that this means inpatient hospitalization, life-threatening, results in death or permanent disability, we have 4 of those in the inoculation arm versus 1 in the placebo arm. And how many patients or how many children that you know where you take 1,000 children and you treat them, that you would allow 4 of them to have inpatient hospitalization, a life-threatening event, results in death or permanent disability if they're at no risk of severe disease. So again, because this is descriptive statistics, we can't ascribe cause to the inoculation and say that it is causing harm. However, it certainly looks like the trend is in the similar direction at 2 months as the data was for the adults at 6 months. I'm just going to move very quickly to our wee ones, the 5 to 11-year-olds who are being inoculated. Again, no risk of severe disease, no episodes of severe disease, slight differences in the number of actual COVID positive, symptomatic COVID positive events or cases here. But we can see again here that in terms of any adverse event, it's 46 versus 16. So they're actually getting COVID-like symptoms more in the inoculation group than we are in the placebo group. And just on that note, what we would say if we started to see something like this, where the symptoms were similar in both groups, and you're getting more in the inoculation group than you would in the placebo group, we would begin to say, how rigorous or how reliable is the actual test that you're using to ascribe efficacy. I'm just going to whisk past this. This is the whistleblower report from the BMJ that basically questioned the integrity of the data that was going into this. Our firm is basically questioning the reporting of the data and the emphasis of the data. We definitely noticed some tricks in terms of trying to boost efficacy and minimize safety by underreporting, and also by using, considering one was relative risk reduction, and then what they did with safety was they buried it in the supplements and they considered it as percentages. And the other thing too that we look at when we see this type of reporting is we immediately go to say, what are the conflicts of interest? Who conducted the trial and who wrote up the report? Because the conclusions for this particular report were that the inoculation was both safe and effective. When we took a closer look at, and with no concerns related safety, no new safety concerns, I believe is how they actually phrased it in the 6-month trial. And we would not agree with that conclusion whatsoever based on our analysis. And therefore we go to look at conflicts of interest. And it is notable that there were conflicts of interest, significant conflicts of interest in the majority of people who were involved in this trial, notably employment and stock for the corresponding author, the last author, and notably the 2 BioNTech founders who basically have earned $9 billion at the time when we made, I think this was made early in the fall of last year. So again, have made incredible amounts of money based on this particular report. So we're trusting these people with these incredible conflicts of interest. And again, even the senior author, the lead author, S.J. Thomas, has had conflicts of interest. They've experienced grant or consultancy or clinical trial development. And notably also, and I just discovered this recently, is after the publication of this particular trial result, which basically boosted the Pfizer stock prices, the CEO of Pfizer divested of his stocks. And again, this is pure speculation, but if I were an insider and I knew the safety data, and I knew the underpinnings of this safety data and what had gone on behind the scenes, then I would probably be concerned about people eventually finding out about this safety issues and might very well be prone to divest of my stocks as well. So my position is such that this trial should have never been passed, that there were issues of I can't, again, it's hard to ascribe causality, but definitely there were tendencies towards overemphasizing benefits and minimizing risk, both in the manner in which that it was unblinded and crossed over, the short duration of trial, the short monitoring periods for the safety results, the questionable test used for efficacy, and again, the emphasis on clinical benefit without really taking a very close look at the safety. And I would have loved to see all of the the unsolicited adverse events to see what kind of adverse events they were reported. But again, all that they allowed us to see was COVID-like symptoms in both groups. And so again, I would say that that's an obfuscation of safety data and that that's minimizing the representation. So it would be difficult for me to understand how, based on the reporting of this particular trial, anybody could possibly say that they had informed consent when they agreed to the inoculation. Thank you very much.